Very good morning, students. In this video, we are going to discuss about biocatalysts, that is enzymes. Biocatalysts are the substance present in living organism, and catalysts are the substance which increase the rate of reaction or alter the rate of reaction. So biocatalysts are the substance which are present in living organism and used to increase the rate of reaction. The term catalyst comes from the term yeast because first enzyme was discovered in yeast. In yeast, since early scientists studied reactions caused by yeast enzyme, and they found that in presence of yeast, any of the chemical reaction can increase. Because biocatalysts are the catalyst. If we are talking in catalyst in chemistry, they generally increase or they may decrease the rate of reaction, but biocatalysts always increase the rate of reaction. Now let's understand the magic of enzymes in our daily life. In this slide, I have done Cadbury gyan. Now, what is the Cadbury gyan? If you have noticed that when in your childhood you or even now when you eat Cadbury or eclairs, these toffees are generally soft in center while they are hard and tough at the covering. इनकी covering hard होती है, बीच में से ये थोड़ी soft होते हैं. It is due to invertase enzyme. Now, what is the role of invertase enzyme here? Toffee means sugar. Which sugar is present? Sucrose is present. Sucrose break down into glucose and fructose in presence of invertase enzyme. No need to go in detail. This is just for your information. Okay. So, invertase enzyme की presence में sucrose break down होता है glucose and fructose में. That's why it becomes, and it is the simplest sugar. That's why it becomes little soft in center. और क्योंकि center में concentration ज़्यादा होती है, इसलिए मुझे ज़्यादा पता चलता है. Now, what is the difference between commercial and natural fruit? This is a natural fruit which I have taken out in home, and this is a commercial fruit which is a market-based fruit juice. I mean Tetra Pak में जो आते हैं, real के fruit juices आते हैं, मैं उन fruit juices की हम बात कर रहे हैं. If you can notice, this fruit juice is comparatively transparent. ये उतना opaque नहीं है. Now, what is the reason for that? When we are making commercial fruit juices, we use an enzyme pectinase that have been used for decades in fruits and vegetable processing during pressing and straining of juices. हम सालों से, decades से vegetables और fruits की processing के लिए, उनको press करने के लिए, उनसे juices को strain करने के लिए हम pectinase enzyme use करते हैं. And that pectinase enzyme impart transparency. Now see in this slide. Now in this slide we can see that when we produce ammonia in laboratory, we need temperature of 400 to 450 degrees Celsius. We need pressure of 200 atmosphere and we need iron catalyst. Now when we produce ammonia gas in liver, we produce ammonia gas in liver by the deamination of protein. That time we cannot have temperature of 400 to 450 degrees Celsius. I mean body के अंदर उतना temperature नहीं होगा ना, और हम ना ही हम इतना atmospheric pressure रख सकते हैं. And how it is possible to produce ammonia gas in liver without such temperature and pressure? This is because of enzymes, that is biocatalyst. Ordination cycle होती है तो उसमें होते हैं use enzymes. Don't worry, these examples are not in your syllabus. This is just for the explanation. Okay. In this slide, you can see that when you combine water plus carbon dioxide, carbonic acid is formed. When we do this exercise or this reaction in laboratory, then 200 molecules of carbonic acids are produced in R. 1 घंटे में कार्बोनिक एसिड के 200 मॉलिक्यूल बनते हैं. And when the same reaction, when we produce carbonic acid in our blood, see, when in blood carbon dioxide is transferred from tissues removed from the body, then carbon dioxide cannot transfer exactly in the same form. Some of the carbon dioxide combine with the form of water and form carbonic acid. So some of the amount is transferred in the form of carbonic acid. So in our body cells, carbonic acid is also produced, but due to presence of biocatalysts, this reaction is very fast. And could you believe that in 1 second, 6 lakhs molecules can be produced of H2CO3? Just see the magic of enzymes. In this slide, you can see enzymes in detergent. Many of the times you have seen in TV and advertisement that कुछ एड्स में आता है कि हमारे 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 डिटर्जेंट में ट्रिपल एंजाइम पावर है और इसके अंदर एंजाइम बेस्ड है. What is this triple enzyme power? This triple enzyme power shows the presence of 3 enzymes: proteases, amylase, mannanase. Why you use detergent? You use detergent to clean your shirt. In what kind of cleaning? When you eat something, तो कुछ खाना गिर गया, पसीना जमा हो गया. When you sit in ground, then sometime dust, sometime that जो grass है उसका कुछ part मतलब उसके impressions जमा हो गए. You have to remove all this oil, dirt, sweat, impressions of proteins and all. For that enzymes are required. उनको तोड़ना पड़ेगा ना, साफ करना, उनको decompose करना पड़ेगा. For that enzymes are required. So when we have proteases enzyme which work against protein based stains such as grass and blood. यदि आपके शर्ट के ऊपर ग्रास का, ब्लड का निशान है, वो एक प्रोटीन है. उनको डिकम्पोज करने के लिए प्रोटीएजेस चाहिए. We have some amylase also in detergents. These amylase works against starchy stains. चॉकलेट का निशान है, खाने का निशान है, टोमेटो सॉस का निशान है. Mannanase, it works against gyan, which is used as thickener in many foods such as ice cream, barbecue sauce, salad dressing, because it is very sticky. उसको भी decompose करने के लिए some enzymes are required in detergents along with the detergent's own chemical. Okay. So in all this slide, you have noticed that enzymes are not only required in our body, enzymes works in our entire life. Now how enzymes are work? Enzymes work as law and key method. Like for every one law, there is a one specific key. In the same way, for every enzyme or for every food item, there is a specific enzyme. When this specific enzyme combined with the food item, ये इसका lock है, ये key है, they combine and form enzyme food complex. And when this enzyme food complex forms, then this enzyme break down these food items and then digestion completed. Enzymes are not only required for digestion, they are required for respiration also. They are required for other metabolic activities also. Okay. So हर enzyme को हम biocatalyst कहते हैं. That is chemical substance present in the body which greatly speed up almost million times the rate of physiochemical reaction. And at the end of the reaction, the enzyme remains unchanged and separate from the product of the reaction. ये देखिए, ये लास्ट में unchanged ही रहा है और अलग हो गया है. Clear? Without enzymes, reaction would occur very, very slowly. So, ना enzymes है, catalyst है, they are like our parents. If parents are not at home, then completion of homework is very slow. And when parents are at home, then completion of homework is very fast. This is just for an example. ऐसा होता भी है, वैसा तो नहीं भी होता है. Characteristics of enzymes. Now let's understand the certain characteristics of enzymes. All enzymes are proteinaceous. हर enzyme एक protein है. But all proteins are not enzymes. सारे protein enzyme नहीं है. But सारे enzymes protein. They destroyed on heating. गर्म करने पर या बहुत ज़्यादा टेम्परेचर पर एंजाइम डिस्ट्रॉय हो जाता है. When our body temperature raised very high, if our body temperature is going beyond 106 degree Fahrenheit, then it has become very critical. And if our body temperature raised by 108 degree Fahrenheit, that is 42 degrees Celsius, at that temperature, our enzymes kill. And if the enzymes kill, we are not able to do any metabolic activities and we may die or our brain cells can damage. So at very high temperature, enzymes are killed. Enzymes are specific in their action. A particular enzyme acts only on one substance. वो एक particular substance पर ही काम कर सकता है. And always forms the same end product. और हमेशा एक ही तरीके का end product बनाए. Look here. This is a substrate. This is an enzyme. They combine and form enzyme substrate complex. Then they form a new product and they decompose into two different parts. So here the digestion or the reaction is complete. Enzyme remains unchanged, वो as it is ही रहा है. Enzymes can be reused since they are regenerated at the end of the reaction. इनको हम reuse भी कर सकते हैं. Enzyme acts on a specific pH. For example, pepsin enzyme acts on acidic pH. That's why along with the pepsin, HCl is present in stomach to provide that acidic nature. Trypsin can act in alkaline, यानी कि वो beyond the 7 pH पे काम करता है. Enzymes speed up the reaction and they can speed up the reaction by millions of times. We have seen in one of the reaction that the ammonia formation in our body, it increases millions times. 6 lakh molecules per second बनते हैं और बाहर लेबोरेटरी में 200 molecules per hour. तो आप देख सकते हैं. So the important question is how enzymes work. Actually, enzymes reduce the activation energy. What is the activation energy? For the completion of any of the reaction, a particular energy is required. And that particular energy is the activation energy of a specific substance. In presence of enzyme, this activation energy reduces. And because activation energy is less, the reaction can complete it easily and within less time. Now, what are the uses of enzymes? Already we have discussed. Enzymes are used in detergents. All detergent does not contain enzymes, but some detergents contain enzymes that break down protein matter, which is deposited as a perspiration, or the food items, or the oil dirt that can be decomposed easily if the enzymes are present in detergents. Enzymes are used in making of antibiotics, beer, bread, cheese, vinegar, etc. During the formation of antibiotics, beer, bread, cheese, enzymes can speed up the reaction. Enzymes can perform the reaction easily. Enzymes are used in diagnosis of certain diseases. They are used in diagnosis of anemia, जो आयरन की कमी से होता है, ल्यूकेमिया, ब्लड कैंसर है, हार्ट एंड लिवर एलिमेंट्स, हार्ट और लिवर से रिलेटेड डिजीजेस में, they are used to diagnose the amount of various enzymes present in blood and other fluids. इससे हम पता करते हैं कि कौन कौन से एंजाइम्स हमारे ब्लड में प्रेजेंट हैं और कौन से अदर फ्लूइड्स में प्रेजेंट हैं, जिससे हमें बॉडी के नेचर का पता चलता है. Enzymes are used to dissolve blood clots. If we have internal blood clotting, कभी हम गिर जाते हैं तो आपने देखा होगा कि इंटरनली कई बार ब्लड क्लॉट कर जाता है. To dissolve that blood clot, enzymes are required. So we have seen that enzymes are very important in our metabolic activities, in our daily application. Now here is a list of certain enzymes which are required in digestion. In mouth, saliva is there. An enzyme present in saliva is ptyalin or salivary amylase, which works on substrate starches. Substrate क्या होता है? Substance जिसके ऊपर हम काम करते हैं. And it form maltose. Esophagus, there is no secretion, no enzyme secreted. In stomach, secretion called is gastric juices and hydrochloric acid. Enzymes are absent in adults and renal in children. Please note, hydrochloric acid is not an enzyme. Now, pepsin is used to digest protein and it converts protein into proteoses and peptones. Renin converts milk caseinosin into casein. In duodenum, the secretion is bile juice, which comes from gallbladder. There is no enzyme. Bile juice is not an enzyme. It is work on fats. It emulsifies fat into fatty acid and glycerol. So here we have pancreatic juices from pancreas. They release from pancreas but works in duodenum. Enzymes present are amylopsin for the digestion of starch into maltose, trypsin for the digestion of protein into proteoses, peptones, peptides, amino acids, etc. Strepsin for the digestion of lipase or emulsification of fat into fatty acid and glycerol. Now, there are certain enzymes which are present in ileum also. In ileum, intestinal juices are secreted, and intestinal juices include rhabdin or peptidase, which helps in digestion of proteins and peptides and convert proteins and peptides into amino acids. Here we have maltase, which helps in digestion of maltose into glucose. Here we have sucrase or invertase, which helps in digestion of sucrose and convert it into glucose and fructose. We have already discussed this enzyme in Acleus. Lactase, it helps in digestion of lactose into glucose and galactose. Lactase is present in milk. In colon, there is no secretion, no digestion, no enzyme at all. Only process is it absorbs water and minerals from the undigested food. Now finally, in rectum, no secretion, no enzymes, no substrate. Only undigested food is stored, that is temporarily, that you have to mention if we are asking this question. Temporarily, it's stored undigested food. Okay, and then this undigested food is released from the intestine. Clear? So by this video, the chapter of digestion is complete. In your book, there is a one question that at what pH is the activity of enzyme is highest. So इस तरीके के questions हम कैसे कर पाएंगे? आपको यदि कोई diagram दिया हुआ है, वहाँ पे pH range दिया होगा, उसके accordingly we can do these questions. Here I don't have picture, so I will discuss later on this question. Okay. Now revise this entire topic from your book do all question answers. Still, if you people have doubt, please ask your doubt in comment section and keep learning, keep doing. Till then, take care. Goodbye.